Thanks for waiting, everyone. We're just watching everybody just join in, and then we'll start in about 20 seconds or so. Wonderful to see such an eclectic mix of people. So we're at 37 attendees right this minute. Okay, fantastic. So I'll make a start. So welcome to this webinar. My name's Anna Middleton and I'm Head of Society and Ethics Research at the Wellcome Genome Campus in Cambridge. And we're really delighted to be communicating with you all through coronavirus via these little webinars that we've been putting on periodically. And today we've got Professor Christine Patch, and we're so chuffed that Chris works with us one day a week and the rest of the time works with Genomics England. I've known Chris a very long time. She's a really experienced genetic counsellor, but more than that, she's very experienced and very has a lot of practical experience in the creation, the construction, the delivery of genomic medicine services. So she's a practitioner. And so she's going to be talking to us today about consent, not just in a clinical setting or a research setting, but both together. And it's not even from a theoretical perspective, it's from a very practical perspective about delivering a consent process through the 100,000 Genomes Project. So I'll hand over to Chris. What we're going to do is, if you have any questions, please pop them in the chat line, and I'll collate those through the presentation while Chris is talking, and then I'll pull these together for Chris at the end. Chris's internet connection is a little bit glitchy, and so we'll start off with her on video, and fingers crossed we carry on video. We've got someone else behind the scenes, the lovely Emma, who will be moving the slides forward for us. We may just go to audio for her if the video doesn't work, and if things just completely collapse, then we may have to reschedule, but we'll just, you know, do what we can to keep going. So I'll hand over to Chris. Thank you.
So thank you, Anna, and in fact you've done all the introduction for me. That's really great. And the main point I think I wanted to make is that my background is in sort of the doing of genomics. And so, of course, the academic discussions about consent, legal discussions about consent are vitally important in how we understand what it is. But actually, I would argue that the doing of consent has an ethical component as well. So next slide, please. If you could go back one, please, Emma. So I just, lovely Laura in, our, in the Society and Ethics Research Group found this cartoon for me. And I think it's obviously an exaggeration, but it's not that much of an exaggeration. There are a lot of studies out there that have participant information sheets that are pages and pages long, that have consent forms that are pages and pages long, with lots and lots of statements to tick and lots of signatures. And I think, and my thinking about this is based on Anna Kalukason's work, we really have to think, what is the work of consent? What are we trying to achieve by this work? And the signature on the form, the paperwork, is not what we can rely on to do the work for us. So there are other things that have to happen. So in the 100,000 Genomes Project, next slide, please. This is just the background. The project, I realized there were a number of international people registered, but just for some context, so the 100,000 Genomes Project was a Department of Health and Social Care funded project with funding from the research councils as well. But it was really the initiative of a government department. And the aim was to collect 100,000 human genomes, sequence them, and they were from patients in the NHS, patients either with a rare disease or with cancer, and return information back to the NHS that might influence care. The other very explicit aim was to create systems that could drive forward genomic medicine and drive forward the creation of data resources that could be used for research, but also to deliver value back to the healthcare system. So this is where we are now. We've stopped recruitment. I think 99.9% of the results have gone back. But we are still, the 100,000 Genomes Project is still going on, because those participants who were kind enough to allow the NHS and researchers to access their data are still part of the project. So next slide, please. So I just wanted to thank the participants again, because it's only through their generosity, their commitment. The project, as you can imagine, was a large piece of work with some quite difficult blind alleys along the way, and they've been incredibly patient. And I also like to thank the NHS staff, because it was a huge thing for them to deliver as well. Next slide, please. So I said before that participants in the 100,000 Genomes Project allowed access to their data. So there is a large data resource. And in that data resource, we have the data from the original sequencing, and there are some samples stored as well in a biorepository. We have the primary clinical data, which is the data that was sent to us by the Genomic Medicine centres at the time of recruitment. We have the bioinformatic data from the analysis pipeline, the variant reports, but also the clinical reports that were made and whether the findings were validated within the NHS and reported back to the patient. And we also have secondary clinical data. And participants in the project have agreed that there can be lifelong depositing of this secondary clinical data linked to their genome within the data set. And this is data from places like NHS Digital, Public Health England, and other data resources. And for example, the sort of data, if we think of, there's a data collection called the Hospital Episode Statistics data set. And that's data that's collected from hospitals. It was primarily for management purposes to start off with, management of the NHS. But every time you attend a hospital in the NHS in England, there'll be data deposited in a de-identified form. In fact, it's almost anonymized, that tells us why you were in hospital, or which outpatient clinic you attended, what the diagnosis codes were at that time. So it's an incredible, useful, lifelong data source for the participant, for researchers. And that is also in this data set linked to the genome. And so our participants have given consent for that. So next slide, please. So to take it back a bit, consent, what does it mean? And as I've said before, the ethical, academic, legal discussions are really, really important. But consent means different things in different contexts. And what we're talking about here is consent in the context of clinical care, consent in the context of research. And a big thing that happened during the 100,000 Genomes Project was that GDPR was enacted, and that's the General Data Protection Regulations, the European ones. The UK has signed up to them and has to comply with them through its legislation. So if we think of clinical care, next slide. The consent in clinical care is consent to treatment. That also includes consent to a diagnostic test. So as a clinician, I have to get permission from the patient to do anything to them. If I do something to them without consent, I can be accused of battery, and that would take me to court. Or I can be accused of negligence, and that could take me to court. So I need their consent to do things to them. But the consent is bound by common law. So in the sense of the law, that is reliant on case law. So we're actually really reliant in the clinical setting on professional standards and guidelines. And a recent case that we've discussed in the Society and Ethics Research Group highlights that. So this is a very important case called ABC versus St. George's, and it relates to the clinician's responsibility to engage with the familial implications of genomic medicine. But until that case came to court, there was no case law surrounding that situation. And that's a really important thing to understand in terms of consent in the clinical space. Next block, please. In the research space, there is a much there's a different legal framework. So the Health Research Authority is a statutory body that sets the way and the standards in which research is conducted. And within the health and social care system, you have to have approval from the Health Research Authority to do research on patients or to do research involving sensitive and identifiable data. And so that consent to research comes under a different governance authority in the research space to the consent to treatment in the clinical care space. And then if we think of the use of data, next block please, under the General Data Protection Regulation, there are various lawful bases for processing sensitive data, or for processing data, and they have a category of sensitive data. You can use consent, or you can use any of the others to meet your legal obligations. The problem with consent as conceptualized by GDPR is it is very, very prescriptive, and it doesn't actually fit for what we do in healthcare settings or what we do in research. So in Genomics England, our legal basis for, lawful basis for processing data is legitimate interest. The word consent is used for all those things, but it has different consequences in different settings. And at early discussions, I think the GDPR use of the word consent was really confusing and led to a lot of issues. Next slide, please. So as I said, in the 100,000 Genomes Project, participants gave permission for whole genome sequencing. And I put that here because the whole genome sequencing consent was under research consent. They gave permission for the return of main findings, that is findings related to the reason for recruitment. And they also had an option to give permission or consent to return of a limited list of additional findings. I can answer questions on that, but I won't. It's a sort of, it's not the main purpose. The additional finding discussion is a really difficult, interesting discussion, but it's not the main purpose of this conversation. And we haven't yet returned those results. At the beginning of the project, consent materials were devised and created, and they fitted broadly what was custom and practice. They were quite long. They were, the consent form in particular was quite long, and I'd always knew that it was going to be an iterative process. And in 2016, there was an evaluation project, and we as a result of the findings of that, we created and revised the consent materials. And this was done with much more consultation. It was in collaboration with participants, with recruiters who had experience of the process, and of course, taking the appropriate guidance from independent ethics committees and other organisations. Some of the materials are still available on the website and the links here. If anybody has any specific reasons for asking for any of the materials, we'd be happy to share you with them. But as the project's finished, they're not actually available on the website to avoid confusion for participants and the healthcare system. So next slide, please. So the project report is also available on the website. And as I said before, it included wide stakeholder consultation. Again, the results are not rocket science. They're predictable. But because they were backed up by this consultation, it wasn't just the clinician's point of view or a particular patient representative's point of view or the NHS's point of view or just a participant in the 100,000 genomes point of view. It took in everybody's point of view, and that makes it more powerful. The arguments you can have about some of these very simple things can be quite heated at times. And if you've got consultation, if you've got evidence that this is what makes things work, it becomes much more powerful. You know, they said, let's make it shorter. Let's make it more readable. Let's have less versions of the forms. Let's have a shorter consent form, and in fact, it reduced by a third in size. We also took the opportunity to address issues of accessibility and produced an easy read version for people with intellectual disability. And that was done in partnership with people with intellectual disability through an external agency. And it was really interesting. One has all sorts of preconceptions about these issues, and to actually go out and ask people, it's the only way to do it. So next slide, please. So just some examples of where we ended up with. As you can see, you possibly can't read them, but they're the information sheet for the cancer patients and the rare disease patients, adults with capacity. A lot of the information sheet was about explaining what genome sequencing was. And that's because in this context, the genome sequencing was done under research consent. And so if you didn't want the genome sequence, then you shouldn't take part in the research. It was not done under clinical consent. And I think that's an important thing just to hold on to in the rest of the presentation. So next slide, please. So I'm looking at my slides on another screen, so I'm just checking where I am. Okay. So the other learning from the 100,000 Genome Project is, what was it like? What happened to the health professionals taking the consent? What did the participants think? And I just introduced two very brief quotes from two papers. The first quote, again, is about answering patients' question about genetics to facilitate genome sequencing consent. In the Genomic Medicine Service, genome sequencing will be a technology that's used to do the test. So there's not a choice about that. If you have the test and the technology that is mandated for the test is whole genome sequencing, then your choice is, do I have the test or don't have the test? But it still talks to, in terms of rolling out a genomic medicine service, it talks to the need for workforce development, workforce preparation, and not to forget that. And I think it also talks to the fact that I can argue, and I'll argue it very strongly, that in the context of a diagnostic test, whole genome sequencing should just be seen as a technology. However, we also have to recognize that there is still something different about genome sequencing. There is something different about DNA. And we need to take account of that without over-interpreting it in a way that means we don't do the best test for the patient. The second statement I think is vital to consider, and it is talks to another previous seminar that Richard did, in that this was interviewing participants and essentially asking them what they'd consented to, and many of them didn't really know. They couldn't recall specifics. But they weren't unhappy about that. It wasn't a problem because they trusted. They trusted the people that received their consent, and they trusted the NHS, and they believed that this was worth doing because it might improve care in the future. And we break that trust in what we do at our peril. And I keep saying that every talk I do about these issues, it's absolutely vital that we retain as much of that trust as possible. So next slide, please. So NHS Genomic Medicine Service. This has been a long rollout. It's a very complicated whole system transformation of the NHS genetic laboratory services in England, delayed because of COVID, but now coming is right. It's we're starting again and we are rolling out. So it's been a long time getting from the idea to actually having a real sample from a real patient going through the system end to end with the result going back to the patient. But that is the process of testing that we're at now of the system. So it will be rolled out in terms of happening very soon. It's a national service in England covering all DNA-based testing, including testing of cancers. It operates to common national standards, specifications and protocols, and this includes the systems in place and the materials in place for recording and obtaining consent. So hopefully there will be less variation around the system. There are 7 genomic laboratory hubs been already contracted to deliver the tests. That's across the whole of England. The tests, what tests can be done, are driven through a national test directory, which mandates the technology, the clinical indications. So individual laboratories will not have so much freedom. They will be working essentially as one virtual laboratory for the whole of England. There's one funding source for rare and inherited disease testing, and the explicit aim is also to build a national genomic knowledge base for research and with academic and industry partners, for trial recruitment around the genomic space, but also for clinical use. For example, we all know that individual laboratories use their variant databases to interpret variants. The intention is that the national laboratory will have the same, a national approach to this, so there'll be much, much more relevant and useful information. And all patients where whole genome sequencing is the technology used for the test will be offered the opportunity to allow their data to be accessed through what we're now calling the National Genomic Research Library. So next slide, please. So as I said before, in the clinical space, most of our practice is driven by professional guidelines, and this document is a really important document in the genomic medicine space called Consent and Confidentiality in Genomic Medicine. The latest edition was last year in 2019. And again, I'm indebted to Annika Lucasen's thinking on these subjects. So the process of seeking consent is about ensuring that the person you're taking consent from understands what you're doing to them, what you propose to doing to them, and therefore they can make an autonomous decision about whether to allow that. Documentation is important, but a signature on a form will not necessarily indicate what consent has actually been given. And it's important to hold on to that. Next slide, please. So in the NHS Genomic Medicine Service, we have called it patient choice rather than consent. That's contentious, it's confusing sometimes, but those of us that have been working in the space over about 3 years now feel very strongly that this is clearer, because what you're actually giving the patient is a choice to have the test and a choice to participate in the research offer. And it's envisaged that clinical care and research choice will both be presented as part of the clinical pathway. That doesn't necessarily mean they'll happen together, but they will be part of the clinical pathway. They are separate choices, but they both need to be clear and informed. And initially, it will be only those people having whole genome sequencing uses technology that will be asked about research. In the research offer, simply, it's about access to de-identified data by researchers, but both from for-profit and not-for-profit researchers. That's explicit. Recall for other research opportunities, lifelong collection of other routinely health data, as I've said, and of course, it also includes samples. And there is the option to withdraw. Next slide, please. Those of you in the clinical space, or any of you that have been involved in the many meetings we held a couple of years ago, will know that something that seems very simple is ends up being quite complicated. And what we ended up with eventually, after moving boxes around and having 3 signatures and then 2 signatures and then 1 signature and then 3 again, is this. The intention was to have it completely electronic based. Of course, with the NHS IT systems, that is not the case. So we've ended up with a sort of paper version of a form that can be used electronically. COVID, of course, is now pushing a bulldozer through all these things that we couldn't do. So it's a really interesting time. Essentially, there's 2 pages. So it's called a record of discussion. And the record of discussion relates to both the genomic testing, the actual clinical test, and participation in the National Genomic Research Library. There is one place to identify that you've had the discussion about the genetic test and you agree with it, and also your research decision is recorded. If we go to the next slide, please. This is some statements pulled out from the clinical form. And mandating that there's a sort of record of discussion form for genetic tests, you'd understand was contentious. Of course, this cannot interfere with good clinical care. So if the test needs to be done for clinical reasons, the person doesn't have capacity, you know, they're unconscious, then of course the system has to be designed to let that go through, and we have the discussion later. But it's implicit, and that the results of a test in this national data resource can be used both for the individual patient in front of you and also for members of their family, but that it will be done in a way that protects confidentiality. Again, I keep referring to Annika Lucassen, but really she's done a lot of thinking in this area. In the old days, we used to, if we used a consent form for a genetic test, there'd be an optional box. You know, I agree to the result of my test being used for my family members. It's not a real choice. Most people agree anyway. The few people that disagreed, actually, as a clinician, if it became absolutely essential, then it is possible you could use that test result. There were ways of doing it with protecting confidentiality. And also, I think Annika might argue it's not reasonable. We share our genetic information with our parents and our children. One could argue that our variants are not our individual property. The combination of our variants are, but as a single variant is not my individual property. It belongs to my blood relatives as well. And this is explicit in this statement. So if you really don't want this to happen, then you have to say no to the test. So when we're talking about patient choice, we're talking about choice bundles. These are not optional. This is what, if you've had the test, this is what you've signed up for. The result will be in a national system. And this is explicit. And I think this is where genomics might be being held to a higher bar, but I could see how it's happened. This was the first explicit national database that was being set up post GDPR. The reality is, in the health service, if you have an X-ray, that goes into a national data system and couldn't be accessed from anywhere in the country relevant to your care. And so there is a national record data system for that. We had interviews with the National Data Guardians, a working group on the National Data Guardians office. And, you know, there was a surgeon, a very, you know, straightforward, who just said, we're all fussing too much. She said, if I want to send a test to the States, I just do it. And the question comes back, then does your patient know this DNA has gone to the States? Do they know what's happened to their data? And so I think it's explicit that this is in a national system, and that the research offer will happen. Next page, please. Next slide, please. In the National Genomic Research Library, we did work both in the literature with clinicians and with participants to pull out what was the minimum amount of information to go on this record of discussion form. The patient gets a copy of this form, and it was about unsurprisingly, security, recontact, what's going to happen to my data and sample, where is it going to be stored, and the fact that somebody has the right to withdraw their permission. So not many statements. Next slide, please. But of course, this was the minimal amount of information. And as in the 100,000 Genomes Project, it's essential that there is a backup of access to more layered, in-depth information for people that need it and want it to make their decision. I just put this screenshot up about what was in place for the 100,000 Genomes Project. We are working based on this on materials for the National Genomic Research Library. I need to iterate at the moment, we're on version 0.0. This is an iterative process, and it will happen over time. So next slide, please. And then came the beginning of this year and the world changed. And of course, COVID did delay the start of the Genomic Medicine Service, and everything we were doing was mothballed, essentially. But the work we'd done simplifying the consent process was essential to us working, this is purely in the research phase, with other studies in partnership with them so that whole genome sequencing could be incorporated into this study. I forgot to mention actually, of course, the whole genome sequencing itself is happening at the Genome Campus. And the first partnership has been with the Genomic study. It's a study that was originally only in intensive care. And we are doing we're storing and doing the whole genome sequencing and maintaining the research environment now for that study with our linkage to longitudinal life course data. And we may be co-recruiting with other studies. And that's very exciting, actually, but it would not have happened if we hadn't already done a lot of the work and learnt from the 100,000 Genomes Project and our developing model to the Genomic Medicine Service. Next slide, please. This is my final slide. So to summarize, none of this, as I said before, is rocket science. And in the patient choice world, consent world, of course, the patient participant's choice has to be free and informed. There has to be enough information, both for the clinical space and the research space, available for them to access to make the choice. Choice must be recorded and complied with. This few words actually is emerged as some of the most complex part of doing consent. In that, if you think about it, we have patients that are in the NHS for their lives. We have participants in our various cohorts in the study for their lives, and we have to maintain a record of their consent choice at a particular time in that data center, and we have to comply with it. And that designing a system to do that is actually a really, really difficult thing. But if we don't do that, then we are not being ethical, is my argument. If we don't regard the system design as part of the ethical and governance component of our research project, we are not being ethical. So it is much more than just a signature on a piece of paper locked away in a filing cabinet somewhere that's inaccessible. So thank you. I'm really happy to answer questions, and here we go.
Wonderful. Thank you so much, Chris. And the video survived, which is
good.
So we've got 3 really nice questions, and so if anybody else wants to ask some questions, please do put them in the Q&A section and everybody can see them. And Lauren, I wonder if you might be able to put into the Q&A section a link to our website, because Chris's talk has been recorded, and so if anybody wants to share this or check up anything from it later, then the recording will be up on our website, so you can access that. Right, okay, so one of the messages that's come privately to us is, I'm interested to know what withdrawal actually means. Digital data, sequence and text, physical DNA samples, or both?
Okay, I meant to say this at the beginning of the talk. Of course, any opinion I express is mine and not the opinion of any of the organisations I've been associated with. In terms of withdrawal, so for the 100,000 Genomes Project, we have 2 types of withdrawal: full withdrawal and partial withdrawal. Partial withdrawal is, I don't want to be contacted again, I don't want anything more to do with you, but I'm very happy for the data that you have to be continued in the research environment, and I'm happy for you to continue to collect new data, but I just don't want anything more to do with this personally. So I don't want to be recontacted for research. I don't want to be recontacted for more samples or more data. Full withdrawal, the sample is destroyed. There is again no contact. The data is put beyond use. The complexity with this clinical research hybrid nature is if some of the data will have been used to generate a clinical result which goes back to the participant. And that, for audit purposes and for governance purposes, you need to keep, but you collect, but it's your data is put beyond use, so is not accessible by researchers. No new data will be collected. Of course, you can't withdraw data from all the research projects it may have been in, and that's explicit in the information people are given. So I hope that's clear enough.
So if somebody consents and has the clinical test, the test is done, and then they request to withdraw, will that clinical result still go back to the referring clinician and be available for relatives?
Yeah, this was very a very difficult discussion actually at the beginning of the project. Yes, and again, this is some of the complexity of a hybrid sort of, you know, where you're directly trying, and I'm talking about 100K, of course, we're directly trying to give a clinical result back. Yes, so what was returned in the 100K was a preliminary analysis. So into the genetics labs, they got essentially tiered a partially interpreted genome, so there were suggestions for pathogenic variants, as it were, related to the condition they were referred for. But it was up to the lab to do that final variant classification and issue report. The result of the first analysis, yeah, we did put that back into the NHS on the basis that it then became the clinical team's decision about whether they wanted to give this back, given that there was an expectation there would be a clinical result back. Does that make sense?
Yes, that's really interesting. So the proband,
but they wouldn't get reanalysis. People wouldn't get reanalysis.
But the proband themselves may have changed their mind and didn't want a clinical result, but a result still may go back and be available for relatives.
Well, that's difficult.
So that's the clinical team, I guess.
Yeah, because the clinical team could choose not to do any further work on the analysis that was returned to them, and in which case there would be no, in a sense, laboratory result.
Yeah, yeah. And with the case law now in the ABC case, the recognition that family has rights to data, even if somebody explicitly refuses to share it with them.
Yeah, and that I think is when you're setting up these systems to actually separate out your responsibilities as a researcher and your responsibilities as a clinical team.
Mm.
So at that time, the pipeline for Genomics England did not deliver clinical results. It delivered potential variants. And then it was the clinical team's responsibility. They could go back and interrogate that data themselves.
Yeah, so then it becomes.
But that's, yeah. In the new world, with the Genomic Medicine Service, it becomes even more complex because essentially in terms of the clinical data coming in, well, it becomes complex because the system operates both as a clinical system and as a research system.
Mm.
So that's even more complex, but it's simpler in some ways because it's very simple. The clinical space is clearly demarcated and the research space is clearly demarcated. Yeah. Virtually.
Okay, some lovely other questions. So how do patients not giving consent for additional use of their data, for example, in the National Genomic Library, how do they verify that this has been honoured?
We will have to have governance systems to, the data is very clearly demarcated, that's one thing. So it's only if and this is why I'm saying the system has to be built to have an accurate recording of choice. So if you say no to the research offer, your data none of your data is ever made accessible to researchers. It's just not in that research environment.
And you can't say no after you've already said yes to the research?
You can say no. Yeah, you can say no at any time.
Any time.
Yeah, yeah.
But then, how would a person check that their data?
Yeah. So what the system needs to be built is so that we can monitor that. So if the person wanted to check, I don't exactly know how you're going to do it, but we're certainly building it so that if a query came in and somebody said, I want to check I'm not in this data resource, we will be able to tell them yes or no.
This chimes with what you said at the beginning, that the pipeline is part of the ethical process, isn't it? The bioinformaticians that are designing the algorithms are part of, knowing about the ethical decision making.
Yes. Yeah. Yeah. And so, for example, in the 100K, for the additional findings, we need to know whether somebody said yes or no to them. Because if they said no, then they shouldn't go into the pipeline for the analysis. Mm. If they said yes, then they do go into the pipeline for analysis. Yeah. And this is where it's been so interesting, that the 100,000 Genomes Project was not a finished project. It was a piece of building work. Mm. But it was necessary, I think, to do to build what we got now. And even what we're doing now, we're still building and learning.
Mm. Absolutely. Okay, so other questions. Let's have a look. So can a participant request to get a copy of their whole genome sequence data?
Yes, they can. And if you are a participant and wanted you to do that, you need to you just email us with a subject access request. And if you go on the website, the actual process, it depends what, I mean, there is a question of what a person might actually want. They may want more clinical input. That's very frequent. Or they may want to take their genome data and have it analyzed somewhere else, but without necessarily having an appreciation of the complexity of that. But of course, at the end of the point, you know, once you've been through the process, yes, you can. I couldn't tell you how many people have actually done that. I guess that's going to be a freedom of information request at some stage, but we'll see.
Yeah.
Yeah.
Okay, so someone has written, this is a bit controversial, I like this, it's fair to say the real message is the only choice for patients is not to even get involved with the whole genome sequencing testing in the first place, and that's a question. So I think this relates to the points you were making about the option bundles. The bundles are quite large, aren't they? So you opt into a whole bundle and you may not agree with some bits of it and you can't opt out.
And this is why I think one has to, whole genome sequencing is a technology that's implied in particular situations and particular contexts. And I think one has to separate that out from just because you're having a whole genome sequence means that all this other stuff can be done with it, but in the context of its use as a technology in a diagnostic setting, it's not done with it. This might be a wrong analogy, but for example, if I have acute back, you know, if I'm referred because for an investigation for, I don't know, acute back pain, and I'm referred for an MRI, I don't question that. I don't say, well, actually, I don't want an MRI, I just want an X-ray. And if I said that, they would say, well, why? Because it's the worst, it's not a good enough test. You know, if you have a child with a complex, potentially genetic syndrome, then whole genome sequencing probably might well be the best technology to use to get you the answer quickest.
And that, I mean, and that's the focus.
Yes, and that's the focus. And you don't, and you don't, your pipeline for analysis is set up to look at the bits that are relevant to the question that's being answered and not look at the rest.
Yeah, but
filter the rest out.
The bundle of what goes together within the option of consent includes things like I consent for this to be shared with my relatives in as sensitive a way as possible and confidentially, you know, we're not going tracking down people, but it's just that it's there for others. So if you actively do not want that, in a way you're not consenting to have the test.
And that's not related to whole genome sequencing. You know, if you have your BRCA genes looked at, BRCA1 and 2, and they find a pathogenic variant, and then your cousin goes, you have that done in Exeter, your cousin goes to Leeds and says, I have heard that there's a BRCA gene in my family, I want to test. The lab doesn't need to know anything about the person who had the original test, but they need to know the variant to get a test that's as predictive as possible in their patient.
Absolutely. Right, okay, another question. What happens if a participant dies?
So if a participant dies, I don't know what you mean in what sense. If a participant dies, their data's in the clinical setting, their data will still be in the clinical space. In the research setting, their data will still be in the research space. But of course, they are now deceased, so they can't withdraw. I guess if the relatives were really strong about it, or were really convinced, we would obviously, ethically and morally, you have to consider a request from the family, I think. But the data's just there.
I mean, you mean ethically and morally if they wanted to withdraw the data, but if they wanted access to the data?
It's a really interesting question. I don't have an answer. It's a really interesting question to consider. Really interesting question. And that's almost a legal question as well as a moral and ethical. It is a legal question as well, a moral and ethical one. There would be your routinely collected health data obviously there's a time lag, but you know, if you're no longer here, then there is no more routinely collected data to collect.
Yeah. Okay, so a couple of other, so we've got here, Wales has introduced whole genome sequencing for all newborns, the first in the UK. Is this initiative likely to be extended throughout the UK? Well, yes, it's happening in lots of places. So yeah, over to you.
Yes, it is. And certainly, so all newborns at the moment, because the whole genome sequence, because the GMS wasn't fully set up with the end-to-end pathways for sampling, but this is a clinical issue. Whole exome sequencing is available at the moment in England for all newborns if indicated. And that will move to whole genome sequence once the systems have been tested properly and worked through. It's part of the test directory.
Okay. Sorry, I was reading the next question as, as you were answering that. I knew what the answer would be. So this is quite a long question. I'm trying to sort of skim it down a bit. So this is about the bundling and the participation in research, and then the introduction of the sharing of data between non-profit and for-profit institutions and researchers, which is clearly absolutely paramount for medical medicine development. But when doing that, it says for profit analysis or for not-profit research, there's been research on what people expect or feel about that. So people do have different preferences, and we've done research on that, as you know. But it's more convenient for the institution not to have an obligation to patients to distinguish, absolutely. So this creates a power imbalance, differential, absolutely it does. So the question is, does the power imbalance the institution has factor into the discussions enough? Oh, that's a good ethics question.
So I would argue, from a different point of view, is actually at the moment, we don't have a way to do that. The complexity of recording choice over a long-term cohort study means that personally, I think at the moment that sort of having very complex choice menus means we can't enact them. So I would argue that's unethical. If once the systems mature and develop, if that became possible, then it becomes possible to do, and it's a discussion to have and a choice to offer. But at the moment, we can't offer that choice because the systems don't allow us. But also, and this might be contentious, we can't realistically offer that choice because it's very, very complex. How do you define for profit? How do you define not for profit? As an academic researcher, you are constantly asked about your IP of your research. Your university can, sell that IP on. Your research may be in partnership with a pharma company. You may say, it's incredibly complex to do properly. So I would say that actually to pretend you're doing it makes it convenient for the institution and individuals, and you can hide behind it. To do it properly is incredibly difficult, and we are being transparent about that, which I would say adds to the autonomy of the choice that the person's making, in that you're being transparent. And then it's their choice. And if they fundamentally don't want this, then they should not allow their data to be in the research environment.
It's messy, isn't it? It's very messy, and this idea that, for-profit bad, non-profit good halos around our heads is nonsense, isn't it? Because
I personally think
as
so,
non-profit researchers, we all have personal benefit from being able to access data sets, whether it's the grants that we get or the papers we can write or the of our careers. And so it may not be in profits for shareholders, we do still personally benefit. And so do clinicians as well, when they can access the database to figure out a variant for a patient in front of them. They get the kudos of getting a diagnosis quicker than their colleagues. They get publications out of it. They get, more patients through the clinic. There's loads of, it's kind of messy. It's not a good, evil thing. We need to be in this partnership together.
And if one works with patient advocacy groups from the rare disease community, and particularly, but also the cancer community, they recognise that advances in treatments and therapeutics and prognostics would not happen without engagement with the for-profit sector. What is really important, however, is where the benefits go.
Yeah.
So it's not the nature of the researchers per se, it's where the benefits go to the people that have allowed their data to be used.
Yeah, and there's quite a lot of discussion about reciprocity, isn't there? And
yeah,
genuine benefit sharing and what that looks like.
Yeah,
yeah,
yeah, and I have done a little bit of work in the last couple of years with Eurodis, the European Rare Disease Patients Organisation, and they are very, very clear that they need to partner with pharma, but that they are also clear that that's not just a one-way partnership.
Yeah.
It's a two-way partnership.
Yeah, absolutely. Yeah. Okay, we've got a couple of coronavirus questions, which I'll just lump together. Yeah, so lovely start. This may be off topic, no, but people being tested for COVID, can their genome be taken and stored at the same time? If so, it worries me that it could break the importance of trust. So you mean the host genome being
yeah.
at the same time as the virus?
Yeah.
Genome?
No, it can't without your consent. So there will be initiatives coming along where somebody has had a positive test for COVID will be asked if they're willing to take part in the host genome sequencing study, but their host genome will not be sequenced without their consent because it's not part of their clinical care.
Yeah.
And you also need a whole blood sample, so just a spit test or a swab or a serum sample. You couldn't do whole genome sequencing from anyway, but if it does happen, then it's completely unethical. It's not by us at all. Your whole genome can only be sequenced with your consent. Your viral genome will be sequenced though, because that's part of the testing.
Yeah. So I suppose what you're saying is it couldn't be sequenced at scale that you're talking about. I mean, in a research setting, you could sequence a saliva sample, but not at scale in a clinic.
Yeah, and it doesn't generate a good enough sample to do the in-depth type of analysis that people want to do on this.
Yeah. Oh, right. Okay. We've got one of, I knew this would come up at some point about VCF files.
Oh, God. This is where you have to remember that I'm not a bioinformatician nor a molecular scientist. So my level of explanation, particularly given this is the Genome Campus, will possibly not be enough, and I may have to think about this. But anyway, go for it.
Oh, I love this. So where consent is withdrawn, what happens to data included in large multi-sampled data sets? An example would be a big joint call VCF file where it's possible to see the data for each individual sample, but removing a single one would involve having to rebuild the entire file and possibly changing results with the downstream analysis. Yeah, okay.
Exactly. I can't talk to the technical aspects of that, but I know I've been in discussions where these things have absolutely been talked about. Yeah, the reality is that we can't, if data has been in and used for research or analysis, as you say, in a large file, you can't track all that on an individual level and you can't remove it.
Yeah.
Which is why we have said we will put data beyond use, and we've been explicit in the material. The whole data resource in the terms of the lifelong collection of data is refreshed every quarter. So the new data, that after that refresh, the data resource available for a new study won't include the people that are then withdrawn or lost capacity and haven't had their consent status updated, or withdrawn is the main reason. Okay. Does that make sense?
Yes. Yeah. I think you'd covered this earlier as well. And then this is the last question. I'm sorry we've not been able to answer every question because there's actually quite a lot here that have come to us privately also on the group. But last question before we finish. So are the police allowed to look inside the genomic database?
No, we have an agreement with the Home Office, and this also applies to insurance industry and marketing, that speculative searches are not allowed. So the police can't look inside the data set. Of course, if somebody got a court order, then it would have to be considered, but we would involve lawyers and it would be taken very seriously. Yeah, and certainly speculative searches are not allowed, and we have agreement with that with the Home Office and the Yeah, the Home Office.
Okay.
And insurance companies don't have access to the data either for individual level risk setting or anything like that.
Fantastic. Okay, so I think we're near the end there. So if anybody wants to rewatch this lovely seminar from Chris, it'll be on our website. Lauren's put the details of our website on the chat bit. So I'll just leave it to thank Chris very much for taking part and sharing her research knowledge with us, and we'll let everybody know when the next time is that we do another webinar. So thank you. Thanks, Chris.
Thank you.
Bye-bye.